Hi everyone, welcome to my talk. My name is Perline Demange and I'm going to present you about our work on the genetic architecture of non-cognitive skills. So first I want to acknowledge that a lot of people made this study possible, particularly Margherita Melenchini, Dan Belsky, Paige Arden and Michel Nivar. We published it earlier this year, so I invite you to have a closer look if you would like to know more. So why were we interested in non-cognitive skills? Well, we are interested in what is affecting how well people do at school or how they go in their studies. So we know that cognitive skills or intelligence, IQ, has a role in educational success. This is reflected, for example, genetically, as we know that cognitive performance has a genetic correlation with educational attainment, so your highest degree obtained of 0.70. But this correlation is not one, so what else is important for educational success? Many individual traits could affect educational attainment, things like your motivation to learn, your curiosity, your social skills, and so on. And these psychological processes have been named according to what they are not, non-cognitive skills. So from twin research, we know that many of these non-cognitive skills are moderately irritable, which means that difference in people's DNA contribute to their observed difference in non-cognitive skills. So we thought, wouldn't it be nice to know more about the genetics of non-cognitive skills? Knowing about the genetic variants associated with behavioral traits, so for us non-cognitive skills is allowing us to do a bunch of things, and for example, allowing us to investigate further and understand the biology of non-cognitive skills, which genes are important and where are these genes expressed. It's also giving us tools to investigate what are the important non-cognitive skills for education or they differ from cognitive skills using genetic correlation or polygenic score analysis, for example. It can help us investigate causal mechanism in education by controlling for the gene-environment interplay, and eventually it could help us understand the mechanism behind correlation between education and other behavioral and health traits. So the standard way of looking at the genetic of a trait is to run a genome-wide association study, or GWAS. GWAS is an hypothesis-free test of hundreds of thousands of genetic variants that we call SNP that are scattered through the genome. And the GWAS is identifying which ones are associated with individual difference in a given trait. So results are usually represented with a Manhattan plot, which you can see here with all genetic variants on the x-axis ranked within chromosome and the significance of the association with the trait in the y-axis. Here you can see the result for the GWAS of cognitive performance and educational attainment run by Lee and colleagues. So we wanted to have a GWAS of non-cognitive skills as well. But there are some issues with running a GWAS of non-cognitive skills. The first problem is that of defining non-cognitive skills. Non-cognitive skills is a tricky phenotype. There are plenty of ways of measuring it and defining it. And studies shows that not all of these measures are equal. So for example, a literature review pointed out that different measures show considerable heterogeneity in their association with education achievement. And others found that non-cognitive measures might have only poor test-retest reliability and some have low irritability. So it's unclear what are non-cognitive skills and which measure would fit better our research question. And then another issue is that GWAS require very large sample size due to the low effect size of each genetic variants and the need for multiple testing corrections. So the problem for non-cognitive skills is that there is only few consistent measures in large cohorts. So to overcome these issues, we decided to use a method called genomic structural equation modeling, which allows us to use summary statistics of existing GWAS and model the relation between the traits and the potential factor underlying them. So concerning the phenotype, we decided to go back to the original definition of non-cognitive skills and define it as all contribution to education attainment that are not cognitive skills. To solve the problem of power, we decided to use the summary statistic of well-powered GWAS. And we use well-powered GWAS of cognitive performance and educational attainment that I showed you earlier. So with genomic SEM, we use these GWAS to separate the genetic variants of educational attainment into two latent components. One first component, which is a cognitive component, explaining the effects shared between educational attainment and cognitive performance, and a non-cognitive component, which corresponds to the effect on educational attainment that are independent of cognitive performance, so which we call NONCOG. So we assure the independence of this COG and NONCOG factor by setting the covariance to 0. And we call this analysis a GWAS-by-subtraction because our non-cognitive factor is all the variance left in educational attainment after subtracting cognitive performance out of it. And GENOMIC is allowing us to get the estimate of the association of each genetic variance with the two latent factors, so that's how we can create a GWAS of the non-cognitive component. So we run our model and we found that COG component explains 43% of the genetic variance captured in the educational GWAS, while the non-COG component explains 57% of the genetic variance of educational GWAS. And this is the GWAS we obtained. So above the red line, you can see all genetic variants that were significantly associated with non-COG, and we found 157 independent significantly associated SNP. So to follow up before to use this new GWAS, we first needed to check that our model was actually working, so how non-COG was associated with education, attainment, and childhood. So we look at the genetic correlation of non-COG and COG with independent GWAS of education and IQ, and we also replicated in polygenic score analysis. And you can see that non-COG, which is in the orange, is significantly associated with education and even more than COG. And for IQ, ideally, we wanted non-COG to not be associated with IQ, because as you can see, non-COG is still significantly associated with IQ, but it's definitely less associated than our COG component. So our GWAS-by-subtraction is a partial success. We do filter out cognition from our non-COG GWAS, but not completely. And one possible reason is that our original cognitive performance, not measuring all of cognitive skills, but I think more importantly, the consequence of that is that if we compare our COG component and our non-COG component in our analysis, we are actually likely to underestimate the difference between these two constructs. So now that we know that our model worked to a certain extent, we can use this GWAS to investigate deeper the genetic architecture of non-COG and investigate the difference between COG and non-COG on different levels: biological level, neurological level, and psychosocial level. So first we look at which biological system matter for non-COG. Using bioinformatic enrichment analysis, we found that only genes that are specifically expressed in the brains are enriched in their effect on COG and non-COG. So the genes associated with non-COG and COG are primarily active in the brain and not other body parts. Then to try to distinguish them at the cell type levels, we looked at the enrichment for genes specifically expressed in certain cell types in the nervous system, using data from 265 nervous cell type in mice and looking at 10% more specific genes for each cell types. And we used a method called stratified score regression and MAGMA. And here you can see the Z-score for the enrichment for COG and non-COG in MAGMA. So each dot here is a cell type specific gen set, and the lines are the significance threshold. So you can see in the top right corner that the genes that are associated with COG and non-COG are specifically active in neurons in the brain. You can also see in this graph that the enrichment for COG and non-COG is correlated. And actually, we didn't find any significant difference in the cell type enrichment between COG and non-COG. And that was a surprise because we defined COG and non-COG to be independent, so as having a genetic correlation of 0. So we decided to check what was happening at the gene level, and we observed that there was no correlation of the association of COG and non-COG with gene expression when we do GWAS. So it seems that COG and non-COG are involving the same cell types in the brain, but the genes that are driving these enrichments are still differentially affecting COG and non-COG. So we could not differentiate non-COG from COG on cell type, so we looked if we could differentiate them at the brain structure level using a GWAS of MRI brain imaging in UK biobank from Zhao and colleague. So here we look at the genetic correlation of total brain volume, total gray matter, and white matter volume. The association for gray and white matter are adjusted for total brain volume. So there seemed to be some difference. You see that the cognitive component is associated with total brain volume, whereas the non-COG components negatively associated with white matter, and none of them are associated with gray matter. So we also looked at the regional level. So here a representation of the association with gray matter volumes on 99 regions. And these associations are also corrected for total brain volumes. So you can see that non-COG was not associated with any regions after FDR correction, but COG was associated with few regions, for example, the fusiform gyrus and the insula. But the correlation with COG or with non-COGs were actually not significantly different from each other. And we look at the correlation with different measure of the integrity of white matter tract measured using diffusion tensor imaging. We look at the association with 5 DTI parameter, but only the mode of anisotropy was associated with COG and non-COG. So here we only represent the mode of anisotropy. So the mode of anisotropy is measuring the shape of diffusion in the white matter tract. So in this graph, you can see that non-COG is associated with a more positive mode of anisotropy in some tracts, so here in red, and so a higher positive mode of anisotropy, which reflects a narrower tubular water diffusion as opposed to a more planar diffusion. And COG was not associated with mode of anisotropy in any tracts, but once again, there was no significant difference between the correlation with COG and non-COG. So it's again difficult to interpret these results. So overall, for the brain structure, it seems that COG is more associated with gray matter and non-COG is more associated with white matter, but the results are really not conclusive. So it would require some replication in bigger GWAS of brain imaging. Then to finally investigate non-COG at the psychosocial level, we look at the genetic correlation of a lot of traits associated to EA using existing GWAS for these traits. So we look at trait linked to SCS, decision-making, risk, health, behavior, and so on. So here I'm just going to go over a few traits and give you some examples. Here are traits related to SCS, and you see the genetic correlation with non-COG in orange and with COG in blue. And the red star is indicating that the genetic correlation is significantly different between COG and non-COG. And as you can see, we found that both COG and non-COG are strongly associated with these traits, but non-COG is also significantly more associated with neighborhood deprivation or longevity than COG. So it supports the idea that the non-cognitive skills that are distinct from cognitive skills are an important contributor to success across the life course. We also look at personality, as these traits are extensively studied in psychology as part of non-cognitive traits, and conscientiousness and openness to experience are hypothesized to be the most important in regard to education. And here you can see that non-COG was most strongly associated with openness to experience, which support this hypothesis. But non-COG was also associated with the other traits, conscientiousness, extraversion, agreeableness, and negatively with neuroticism. And this is an interesting pattern, and it is actually resembling the change that is occurring when young people mature into adults. So with an increase in openness, conscientiousness, agreeableness, and a decrease in neuroticism. And another interesting point is that all 5 personality traits seems important, but all of these correlations are quite weak. So non-cognitive skills are probably not only personality traits. And to finish, some other traits we looked at are psychiatric traits, as mental disorders are usually associated with lower educational achievement phenotypically. So we found surprising results here that non-COG is actually associated with positive or with higher risk for multiple disorders like schizophrenia, bipolar disorder, OCD, and anorexia nervosa. So that was an interesting result. And another interesting result here is that ADHD is not more strongly associated with non-COG than it is with COG. So that might be an indication that non-cognitive skills are not only or not that much about inattention or attention, that they are sometimes fault. So as a conclusion, we show with this study that genomic can be used to conduct GWAS-by-subtraction and create a GWAS of unmeasured phenotype. And here we used it to conduct the first GWAS of non-cognitive aspect of educational attainment. When we investigate the difference between cognitive and non-cognitive factors, we found there's no difference at the level of brain cell type, but there might be a difference at the level of brain structure. We also found significant different genetic correlation with personality and psychiatric traits. And the last thing I want to finish on is that this new GWAS is a useful new tool to continue to investigate non-cognitive skills and education. So to give you some example, in this preprint we just recently updated, we create polygenetic scores of non-cognitive skills and cognitive skills, and we use these to show that cognitive and non-cognitive skills of parents are influencing their children's education achievement above any possible genetic confounding. And in a manuscript that is currently in preparation, Margherita Melenchini is also using these polygenic scores and is showing that the role of heritable non-cognitive skills in educational success is increasing across development, while the role of cognitive skills is constant. And finally, just last week, another preprint was released from another team who used this GWAS method to show that the relationship between psychopathology and cognitive ability is shaped by prenatal neurodevelopmental mechanism, while the relationship with non-cognitive skills is involving postnatal synaptic pathway. So I'm very happy to see that these good results are coming from the use of these GWAS, and that the GWAS can be used to really investigate non-cognitive skills further. So I thank you all for watching, and I hope to see you at the symposium and to be able to answer some of your questions.